Um, let me just introduce the meeting for everyone, and then I will pass it to you. So welcome everyone to the
Julia. Hi everybody.
Hello everyone. We're about to start, so if that's okay, I'm gonna mute everyone for a minute just to introduce Daniel, and then hopefully you can participate definitely in the discussion. Please throw your comments, etc. So let's start for now. And okay, this is the 18th occurrence of the Community of Practice meeting. The agenda for today is that we'll have Daniel Tolhurst from the Roslin Institute, but I will let Daniel to introduce himself. He will be talking about modeling GxE interaction in global plant breeding programs. He is a complex topic, but I think he has tried to simplify it, but still I wanted to allocate a good portion of the time so he can relax and provide all the comments that he has. And then we can participate in a discussion at the end, which I think would be pretty interesting. So please wait till the end. And just a quick reminder about the purpose of this community of practice. You know, this is supposed to be, and we're trying hard for this to be a forum to discuss topics that are related to breeding scheme optimization and germplasm management, provide guidance in the implementation of new ideas, also, you know, be a place where people can share those ideas, new tools, new methods from all the collaborators on the Crops to End Hunger, like CG centers, private industry partners, or excellence in breeding. And just a final comment, but please pay attention to this one. If you want to volunteer to present something that you're currently working on, please send me an email. I think I have talked to you multiple times in your programs. I'm pretty sure because I know you that you are doing very interesting things, whether it's cassava, yam, rice, potatoes, maize, etc. I'm sure that you're doing something very interesting. I have seen it when it comes to cycle time, when it comes to better selection strategies, etc. Please volunteer to participate in this community of practice. Okay, and that's all what I wanted to share. So after this, I will pass it to you, Daniel. I hope you can take from here. Let me know.
Thanks, Eduardo. I'm not sure if you want my video or not. What do you
that's really up to you, depending how good your internet is. Mine is normally really bad here in Texcoco, so that's why I do not activate it. But
okay, well, I'll keep it on, and you can let me know if it sort of affects anything, and I can stop it midway. So I'll just share my screen.
Yeah, please go ahead. Should be a green button there. Yeah, I think something is coming. Yep.
And that should now have worked.
Yeah, that's presenter's mode. That looks good.
Okay, perfect. Firstly, I have to quickly apologize because it seems like my computer also thinks it's quite hot weather and the fan's been running non-stop. So I'll try and speak clearly so you can overhear me over the fan. Thank you for the introductions, Eduardo. Also, thank you very much for the opportunity to speak. I haven't actually joined this forum before, so I'll go through a little bit of an introduction about myself. But as Eduardo has mentioned, I'm sort of, my PhD or my research to date has really been in sort of modeling GxE interaction in plant breeding. In this case, it's sort of global plant breeding because we're talking about sort of Bayer crop science here. So I'll just quickly, like I said, introduce myself. Hold on a sec. Again, because this is the first time sort of I've had an opportunity to meet the people in this group, I just quickly introduce myself. So I graduated in 2014 from Australia at the University of Wollongong. It's just a sort of a small city there on the east coast, a couple of hours south of Sydney over there. And I've graduated from a Bachelor of Medical Mathematics. Essentially, it's a bit of a nice name to, you could either go in and do a postgrad in medicine or applied stats in agriculture and whatnot. And that's sort of where I've headed. I was fortunate enough to get a position at the same university for 5 years. And I worked mostly in the Australian grains industry as a biometrician. And that group was headed by Professor Brian Cullis and Dr. Alison Smith, rather. And so a couple of sort of nice names in the plant breeding biometrics area. So I learned a lot from those two in that 5 years. I was then visited the Roslin Institute at the University of Edinburgh. So I got to meet Dr. John Hickey and Gregor Gorchank, and subsequently I was awarded a PhD studentship in genetics and genomics. And I guess what I've sort of got a bit of a background in biometrics, I'm really now trying to tie that in with the genetics here at Roslin. So that's my PhD, that's sort of from 2019 onwards. But like I said, I've got fortunate enough to have some really good experience in the Australian grains industry there. So I usually like to start my presentations with an acknowledgement. And in this case, it's to acknowledge the other people in this research. So it's a joint collaboration between the Highlander Lab there and Bayer Crop Science. So we've got Gregor, Chris, John and Brian are the main sort of players there in this sort of research. Again, like to thank Bayer for the studentship as well as the use of their data there. And a special mention is that my research till now and moving forward wouldn't be sort of structured the way it is, or I wouldn't have the fundamentals without Brian and Alison as well. So big thanks to them. And we published a paper in 2019 on genomic selection using factor analytic models and all this nice stuff in plant breeding programs. And this has sort of really set me up for where I sort of want to go in the future and whatnot. So now to the actual presentation, just a brief outline of what I'm going to be talking about today. As Eduardo mentioned, I am going to be sort of mentioning or talking about GxE interaction, but I'm really going to do it in a way that is not too technical. It's just more or less from a higher level, sort of speak about what it is, you know, why we need to account for it, but even to a larger extent, why we need to model plant breeding metadata properly. So we have a bit of background, some history, GS, and this is a bit of a controversial point, but why I sort of say, was it a bit of a step backwards for modeling GxE interaction? And I'll explain exactly what I mean there. So please don't just quote me on that statement alone. And what way can we actually move forward with this sort of stuff? So I've just got this general plant breeding scheme from Chris Gaynor's 2017 two-part paper. And there's lots of stuff going on here, but I mean, like we all sort of know that the plant breeding is some sort of 8-year cycle from the initial cross to the release of a variety. And without going into too many details here, what my research or a lot of our research on GxE is mostly to do with the yield trials, and that's where we're looking at the final 4 years essentially. So not particularly interested in any of the crossings or all that. It's really about gauging GxE in the yield trials, like I mentioned. So this figure just sort of highlights that. And what I'm talking about today is really going to be sort of focusing on these later stages. So what are plant breeding yield trials? Well, they're comparative field experiments. I've got a nice photo here from a collaboration back in Australia that I had with the Australian rice breeding program. And so these are actually some rice plots here. What's actually a bit more informative is this next picture. And again, I'm not going to go over it too much because I guess a lot of the people in this forum have seen these sorts of things, field trials, yield trials and whatnot. Essentially what I really wanted to point out here is that like a lot of the programs, the Australian rice breeding program actually has multiple stages within the same fields. And we can see this is indicated by the smaller plots up in the far end of the figure here. Those are the earlier stages. And then we have sort of the late stages down in the foreground here. And if you're wondering why, oh, actually, no, I mentioned it's rice and that gray, that's just the water being flooded in there that you can see there, not sand, obviously. And again, so we have these comparative field experiments where you have these square contiguous arrays of rows and columns, and the plots laid out. And these ones are typically 10 meters long by 2 meters wide. And then the shorter, earlier stages will be about 5 meters long by 2 meters wide. But again, the main point of this is that, you know, we know that we put all the material in the same field. And so this really affects the way that we should be analyzing the data as well. So just to repeat on that, we do have these nice experimental designs, which is a bit of a benefit in plant breeding, where we have this replication of our genotypes. We do use some classical and as well as model-based designs that sort of developed over time. And I've mentioned one sort of the PREP of Brian's from 2006, and that's really essentially what's been rolled out in the Australian plant breeding, and sort of seeing it extending internationally as well. But there's been a lot of research in the experimental designs from Williams and Patterson, etc., from various areas. And what that replication allows us to do is actually partition our additive effects, so our breeding values, as well as all the non-additive variation as well, so epistasis, dominance, etc. And again, this is just another sort of highlight thing that we can be doing this in the analysis. And this is also, I mean, having replication here is where I'm sort of mostly talking about line breeding programs, but it doesn't need to stop there. And what the replication also allows us to do is look at that non-genetic variation in the field. So we have the residual, so what we sort of talk about when we have correlation structures at the plot level, as well as extraneous variation, things like competition, sowing direction, harvesting, serpentine harvesting, all that sort of stuff we can account for in the analysis. And again, I'm mentioning this, another Australian paper. There's a bit of a common theme here, but nonetheless, a lot of the spatial modeling approaches that I advocate and that sort of used widely are these based on this Gilmore, Arthur's paper in 1997. So again, just what we're really highlighting here is that we have these lovely designed experiments, or these field experiments, these yield trials, and we know that there's lots of variation in the field, and everything that we basically structure the way we analyze things is based on how we've designed it, as well as all these peripheral effects. And so that's what you're sort of typically seeing within a trial, within a location. And then what we do when we construct a MET data set is we put multiple trials together. They'll cover multiple locations, years. They may also cover multiple stages, sowing windows, all this sort of stuff. And there's no rule of thumb of how to, you know, construct a MET data set, rather than, you know, it depends on the aims of your research and what you want to get out of it and whether it's appropriate or not. So I've mentioned sort of things here that you can do, combine different years and stages and whatnot, but, you know, obviously only if that's appropriate to do so. And once you construct that MET data set, what the plant breeders really typically want to do is assess the performance of their lines across those different environments and gauge the genotype by environment interaction. So just at a very high level, I talk about genotype by environment interaction. We broadly classify it into two categories. The first, well, apart from that, when we talk about no GxE interaction, is when I have environments 1 and 2, and within those environments, I have the performance of genotypes 1 and 2 here. And since the lines are parallel, there's no interaction. So it's just that old classical interaction plot. And what we can see there is that although the performance increases in environment 2, the rankings don't change. So we basically say that there's no GxE interaction observed in that figure there. And this is actually what we're doing when we were saying that we have genotype main effects, essentially. That there is a difference in upward direction in the performance, but but their rankings don't change, and also the differences between the performance don't change either. That's just changing by different environmental means. In the actual two classifications of GxE that we're interested in, or more so interested in, is what we call scale, or where the genotypes ranks don't cross over. We can see there that the change in performance between the two environments do change, but the difference between the two in environment 2 is larger. So that's what we call a scale GxE interaction. That's still a form of GxE interaction. The other form, which is probably more important to plant breeders, is that crossover GxE interaction, where genotype 1 is better performing in environment 1, but then it switch ranks to genotype 2 being better performing in environment 2. So we have these sort of different things going on, and again, it depends on what you want to get out of your analysis, what type of GxE you want to model, what type of GxE you have in your data, and where it actually your environments are situated. But in my experience, at least back in Australia, is that the crossover interaction is substantial, and scale interaction is also very important in the data sets. We rarely, if ever, actually model anything with a simple main effect on the left-hand side. It just doesn't occur in the data sets that we see. So I guess what I sort of wanted to really, use this talk for, as well as not only introducing a bit of my PhD work, is a bit of more of a discussion points of, where we've come from and where we're going, and raise a bit of discussion in, maybe ruffle some feathers about, where we should be going as well. So, I mean, GxE interaction has either been known for or modeled in one sense for a long time. There's no secret about GxE. You can't really, or at least in Australia, you don't go past talking to a plant breeder without talking about GxE. And so it started off with the initial analysis of the variances, where you had simple main effects in your ANOVA. You then went to things where we try to explain a bit more of the GxE and a bit more information about that GxE, where you got these Finlay-Wilkinson regressions, and which is essentially just a regression on the environmental means. And that sort of showed, well, it became very popular, and it's still used today, these Finlay-Wilkinson regressions, but the problem is that the environmental means rarely actually capture a lot of the GxE in your data. So it's sort of a bit more used as a surrogate for a more complex set of covariates underlying your environment. And then there was also this push at a similar time to getting more interpretation from GxE analysis. You've modeled it in such a way, but what can you actually do with it? As a plant breeder, how do you use it? So then you sort of also went through these PCA type approaches, which was an AMI, where basically you started looking at, or rather visualize the residuals from ANOVA in a PCA type manner. And sort of that was popularized by in 1992 as AMI, like I was saying. So like I said, it's just more that there's, we've come a long way and we're still going. So a bit furthermore, like I said, I wouldn't go past actually mentioning a bit of the Australian work in the factor analytic models by Smith and co. And really what that enables you to do is actually capture a large amount of your GxE interaction. One of the offsets to that is that it's actually used as latent environmental covariates. So it's actually estimated from your data. So this has a downside of plant breeders really can't interpret what these latent factors are that's driving the GxE in their data. So again, you have that trade-off of it will capture a lot of GxE, it'll do a really really good job at fitting the data well. But in terms of, well, what can you get out of it? There's been a lot of research in the last 20 years. Another paper I haven't actually put down here is Brian and Allison's 2018 paper, where they're actually showing a bit better of how you can use this factor analytic model to then make selections. And they've got a selection tools paper out. So I do recommend that one as well. But again, that does not cover the fact that the factors are latent. And then obviously in the realm of genomic selection too, we've got the random regressions, or at least you don't actually need genomic selection for random regressions, but the random regressions on the known environmental covariates. I've got two examples here. There's a couple of others prior to that that's also pretty good, but these are typically involve the stress covariates, which they collate using daily weather observations and whatnot. And that's really enabled plant breeders to get a bit more of a gauge into forward predictions as well, unobserved environments, all this sort of stuff. But it starts with that they can actually make some tangible decisions based on covariates that they can interpret. And so that's sort of an offset to what is happening in the factoriality models. But the big downside for the random regressions is that they don't actually capture a lot of the GxE again. So we sort of go backwards a bit in a way. And so there's no real method to date that allows you to do a bit of both, you know, capture a lot of GxE and get forward predictions and get interpretability. So I'll come back and talk about this again when I speak about a little bit of my PhD research later on. But so we've come a long way with modeling GxE and modeling metadata, and now we sort of hit the genomic selection in 2001. So we have this big push now to use genomic selection for everything. And it was initially developed in animal breeding, and it's been subsequently brought over to plant breeding. And what we've seen, or at least in my experience, is that a lot of the previous methods and the kosher stats methods of analyzing plant breeding metadata sort of went out the window a little bit when this genomic selection was brought in. And I'll explain exactly what I mean there. And a lot of the time there was actually the fundamental differences between the two settings were sort of either, you know, neglected or put as a secondary objective of the research. And we started doing things like two-stage approaches where you would get means in individual environments and then use those means in a second stage and then just use a GBLUP type approach. And we also started forgetting about the non-genetic variation. So I spoke about the spatial modeling, experimental design, all that sort of stuff. So it sort of, yeah, again, took a back seat. And even nowadays, what we see in the way that sort of these things are taught is that, oh, the two-stage approach is a way to go, and really not so. Like with the advances in computing and everything like that, we really should be putting the raw data in a single model, modeling our non-genetic variation, modeling their GxE, you know, with our markers and everything all in a single model. And these sort of two-stage approaches are less so warranted these days, at least. Again, depending on what sort of size data you have. And so another thing that's really become apparent is that when we hit genomic selection was that everyone's sort of become more interested in these, well, there's always been an interest in main effects, but particularly interested in just the main effects, because they were saying, well, we run just a GBLUP and we'll get these main effects. But in fact, there's no reason why you can't get all the effects that you want, interaction effects, etc. And so this is really why I sort of say step backwards for plant breeding. I don't actually mean that we shouldn't do it. I absolutely think that we should be using it, but in an appropriate manner that in our plant breeding setting, and that we shouldn't forget about everything that we've sort of, all the research that has been done over the last 100 years or so to get us to this point, just because genomic selection sort of come along, especially since the genetic variation is typically not the most substantial in field trials, or at least or at least you can't neglect the amount of non-genetic variation there is as well. So that, again, that's sort of what my point is there about that. So having said all that, and hopefully brought up a few discussion points, my research at the moment in my PhD is really following on from sort of the fundamentals I've learned over the last 8 years or so, and taking that forward with genomic selection. So it's why I sort of say this, reintroduce appropriate methods of analysis for plant breeding metadata. So we got things like proper experimental design, construction of metadata sets. And what I'm really going to finish off the talk with today is talking a bit more about how we can use informative and interpretive models for GxE interaction. I'm just going to quickly check the time so I don't, oh, no, I won't.
Yes, we are 30 minutes over the hour, Daniel. So you may want to speed up maybe 15.
No worries. Yeah, fine.
Yep.
All good. Thank you. So what I'm really going to start with a bit of a motivating example. And this is where I've gotten to with the Bay of Cotton breeding program from the US. And on the right-hand side we have this lovely picture of the US states. And what we have here is a late stage program, like I was saying earlier. We have 18 environments that sort of cover the cotton growing regions from the east through to Texas. And those three major growing regions are highlighted with our different symbols here, the north, the south, and then the west over in Texas. And so that really encapsulates the data set that I've been working with initially anyway to develop the models in PhD. So just a bit of a summary here of the phenotypic data. There's a fair bit going on the right-hand side, but I'll sort of highlight the most important parts is simply that we do have those 18 environments covering the 8 states. We have 3 trials within each environment, and those trials are simply just a subset of the total number of genotypes, and those total number of genotypes is 208. So typically we have about 62 or so genotypes in each trial, as well as a few checks. Everything is completely replicated, and then the checks, we have a few more replications because they occur in, like I said, every trial as well. And those 208 genotypes. So everything is about 430 plots, and then you have some missing values due to being lost in the field, etc. But everything's based on similar sort of size designs in each environment. And then we have the yield, which is recorded in bushels per acre, that varies substantially between environments as well as locations and regions, etc. And then I'll just wait for that. And then we have the reliability, or which is just a generalized heritability measure on the right-hand side. So again, that's varying substantially between environments as well as states and regions, etc. So we have a nice structured data set here, but there's a few things, that is going on that needs to be accommodated, which is fine, which I'll talk about in a minute. And now what we do also have is a series of environmental covariate data that's been measured. Initially, about 46 different covariates were measured for each day across a year. So we have daily observations. And what I've done is just subsetted these down to about 18, just for use in this, like I said, in preliminary analysis. And what I basically do is, over those observations over the year, I just take the growing window for the trials in this cotton data set, and we take the covariates according to that growing window. And so again, the main point here is just that we have these covariates based on latitude and longitude. These aren't stress covariates; these are just the raw observations from the weather stations. And we have these differences going from north to south to west. And these are just the simple means of the covariates. So averaged over each of the environments in there, just to give an indication of what's going on here. But again, the main point here is just that we have a series of covariate data that we have. And then to wrap things up in terms of genomic selection, we've got the marker data on 204 of the lines. We have 36,000 SNPs, and we filter those on minor allele frequency and missing values. So we end up retaining about 24,000 SNPs from the analysis. I can't give too much of the details away about how, all the details. It will be covered in the paper, but for now it's sort of confidential, like I was mentioning before. And then I just construct the genomic relationship matrix according to the Van Raden-1 model, which is the centered allelic scores at each marker, and then divided by the marker variances. So accumulating all that, we have a nice little data set going on here. Like I did mention that sort of for global mate breeding programs, in this case, this is just a smaller subset of one of the larger data sets just to get things going. But we have some trial designs, we have different trials within the environments, we have multiple environments, we have multiple states, you know, so there's a large amount of GxE that we'd expect to go on here. We also have the markers, and then on top of that, we have the environmental covariates. So I mentioned before that you have an FA or factor analytic model, which has been quite popular in modeling GxE because it captures a lot of the variation, but it doesn't give you much interpretability in terms of those latent factors. And then on the other side, we have the environmental covariates or random regression approach, which allows you to do forward predictions, interpretability, but it doesn't capture much of the GxE. And so my research has basically developed this new integrated factor analytic linear mix model. It's a bit of a mouthful, but basically it takes the best of both worlds. You get a large amount of GxE that is captured, as well as it being driven by, or at least explained by, the covariates. And so you now enable this GxE to be interpretable, and we can now use factor analytic models for forward predictions, which is great. I'm really only going to touch in the next couple of slides on this interpretability thing, but rather just sort of say we can do everything, all the nice things that we really want to, as well as modeling the non-genetic variation, the experimental design, and all that sort of stuff. So the final couple of slides is just a little bit of the results. And because I didn't want to go too much into the details, being the forum that this is, but please ask questions, and also, like I said, there's a paper coming out very soon to explain everything as well. The best way I like to sort of present it is this figure here. These are what we call the correlation heat maps between environments. So basically how well they are correlated. A correlation of 1, which will give you a red, will basically say the rankings of the genotypes in those two environments are the same. You go all the way down to -1, and that you have the rankings are completely reversed. And then somewhere in the middle, there's some sort of non-similarity between that. And so between the 24 environments in this data set, the figure on the left-hand side was if we were to construct this covariance matrix or correlations between, using environmental covariates alone. And using the environmental covariates alone, we can sort of see that there's a little bit of similarity with the West, within the North, not much within the South, and not much sort of between it either. And this is really my point, in that the covariates don't really capture much of the genetic variation to the extent that we would like. On the right-hand side is basically the new approach, where we use a combination of the environmental covariates as well as the latent factors, and really boosts up those correlations. And it's not like it's just overestimating the correlations or anything like that, but rather it's because it's capturing more of the genotype by environment interaction. And so now we can really see that there's a lot more similarity within the South, a lot more within the West, and then there's also that similarity within the North and the South. Again, the West is a bit of its own sort of little beast out there, in that things are quite differently adapted out there. But the important thing to note here is that we can still put that in the model. We can still estimate all the correlations that we need. We gain strength from having those multiple environments. We have predictions across the data set and whatnot. In this case, it is a fully balanced data set, so everything's nice, but if it's unbalanced, we get those predictions into the environments so that they're not grown in. And the last one is a little bit more technical, but it's just, again, a follow-up of the previous slide, where the last slide really looks at everything from the environments, how well do the environments compare to each other. The other side of GxE is obviously from the variety or the genotype side. And these are just some summary plots here, which I'll go through in a tick. So I mentioned before that the traditional factor analytic model is really sort of popular because it does capture a large amount of the GxE or the genetic variation. And in this case, we fit increasing number of factors and to capture that variation in a bit of a way of like a factor analysis. Well, it is a factor analysis in a bit of a way of like a PCA type thing. And in this case, we fit 4 factors to the data and it captured 75% of the genetic variation. And what I've just done here is partition that genetic variation into the different factors. So we can see that the first factor captures the most, followed by the second, followed by the third, followed by the fourth. And there's all nice little things going on here, like that they're orthogonal and all the rest of it too. So it makes interpretability quite nice. Now, the left-hand figure here is no different to what you would attain from the normal factor analytic model. I mentioned before the selection tools and whatnot of Brian and Alison. So that's very similar to what they were doing. Actually, these figures are based on what they were doing. The difference now is the right-hand side. And the right-hand side is how those different covariates are actually driving those factors or how they're explaining those factors. Now, previous to this approach, all you could really do is get some environmental covariates and back correlate it with your factors. And I've got an example here. Factor 2, which accounts for 16% of the variation. If I was to get the longitude rather, it has a correlation of 0.8 with that factor. And you can see that quite nicely because all the X's which correspond to the Western environments are on the right-hand side of these plots. And all the left-hand side has the north and south. But you can't actually do much besides saying that there's a correlation there. You can't actually utilize it for forward prediction. You can't actually use it to explain what's going on. And now with the right-hand side, with the right-hand side of figures, now we can do that. Now, I'll just take a quick step back and explain exactly what these figures are showing. The purple is a Czech genotype, which I've just called C1, and the blue is a Czech genotype, which I've called C2. Now, the performance in any one of these environments, the environments corresponding to the data points, is equal to the sum of the regression lines, basically, over the 4 factors. And that's why you'll get deviations from the regression line in the first factor. Each subsequent factor is then adjusted for the previous factors so that in the end you end up getting it just along the regression lines there. So basically your performance in any of these environments is equal to the sum performance there, sum performance there, blah, blah, blah. So that's what's going on there. The first factor, like I said, captured the most variation. The regression lines never intersect, so they actually correspond to scale G/E. And then the subsequent factors, they intersect. So they're those crossover G/Es that I was mentioning before. And so I guess that's a bit of the main points that's going on there. Now, with the addition of the environmental covariates, we actually get these factors being driven, like I keep calling driven by the different covariates. And I'll just sort of highlight a couple. One is cloud cover. It's contributing to 0.44 basically of that second factor, 0.3 and negative 0.4 of the fourth factor. And essentially now what breeders can do is start actually interpreting these factors in that manner. And we can see that the environmental covariates basically explain all of the final three factors. On top of that, you can see that there's only about 10% of the first factor being explained by the covariates. That's okay because that first factor is just scale G/E anyway. So that's a generalized main effect going on there. So you're not really needing that for forward predictions anyway. It's all the important stuff is happening in these final 3 factors. So I probably won't delve into too much more. There's a lot going on here, I understand, but I'll leave it for some discussion because I want some time for that too. So I'll just basically finish up with saying that this sort of method has been a good way that we can move forward. I'm not saying it's the only way by any means or anything like that, but it's because it hasn't lost sight or we haven't forgotten about what we've done up to this point in terms of the experimental design, non-genetic variation. We've also got that interpreter model for G/E. I haven't spoken about forward predictions too much, but simply that you can now use factor analytic models to get a prediction of any genotype into any environment, whether it's observed or not. And like I said, the paper is coming out very soon. If you would like any information on the references, please contact me. Other than that, I'm happy to field some questions or discussion or whatnot. I think there was a couple that popped up in the, I'll just open that.
Okay, yeah, thanks, Daniel. That was very interesting. I would say at the beginning, I was getting a bit lost. Now, as you were explaining, I understood what you were behind. No, that's actually looks pretty good. This is great progress. Basically, these new methods are taking us to understand what is really behind what we call GxE and all those latent factors. And I guess, Daniel, maybe I will throw the first question. So hopefully this will trigger the discussion. Let's say that now we understand behind GxE, we start to understand, okay, there's the temperature, or is the cloudy, average cloud out of time, etc. How do you foresee that this information will actually help the plant breeders when it comes, to some of the decisions that they need to take, like selection of parents or advancement of products? What's your think on, or your take on that?
Yeah, I think that's probably a better question for a plant breeder, because I don't advocate myself as a plant breeder per se, but I guess my approach to this is to give them the best information that they can then make their decisions. So I don't want to sort of say something wrong with how they can explicitly use this in terms of, all right, we'd start breeding for cloud. I don't know how you even start doing that. But the way that I would more say, or the way that I'm sort of moving forward with this is that I've sort of spoken about the response of the different genotypes to the different covariates. You may actually have a particular region that you're breeding for, and within that region, you actually observe a range of covariates. What I mean by that is you experience a range of temperatures, you experience a range of cloud cover and whatnot. And what we're really focusing on then is that, given that distribution of your covariates, what are the best genotypes that are adapted to that? And that we can do with this model.
Yep.
And that's more the sort of a way, as well as, yes, you can get a prediction into any environment, but it's more about, you might actually observe a range or distribution of the different covariates in your environments. And then it becomes, well, which ones are best adapted? So if I quickly go back, there may be, obviously our GxE one is better in all of these, but there may be actually areas where the other genotype is the better choice based on the covariates that are observed in that environment, rather than, just trying to disentangle this stuff from a normal factor counting model and say it's only based on what we observe, what was observed in our data set.
Let me actually just throw something there and then pass it to Gary, who's been trying to raise his hand. Because I think what comes to my mind, and probably to many of the people in the audience, is that this way to understand what's behind GxE can actually help a lot to the efforts of defining the TPE, because we normally to define the regions where we should target our efforts, and normally it is difficult to separate that. I mean, what is the good TPE1 or the TPE2, etc. And by understanding that maybe I don't know, the temperature is going to drive this, make this crossover interaction to show up, that can help to understand, okay, then we should be careful when defining the target region or target population of environments, because this could be influence a lot in the crossover interaction. Gary, do you want to say something on that?
Yeah, it's on the same lines. Thank you for the talk, Daniel. You mentioned that you don't want to tell plant breeders how to use this approach, but it's extremely important that biometricians have a clear understanding of the problem that they are advising their clients on. And the principal problem in plant breeding is not to take a bunch of fixed lines and to try to figure out what environment they work in. That's virtually a non-important problem, because the breeding programs at seed companies, extension systems, essentially target breeding efforts, testing efforts, and recommendation efforts at predefined target populations of environments. So your approach looks, helpful to me to define those TPE, but it's very important to put this type of work into a selection theory framework. The real objective of plant breeding is to generate a high rate of genetic gain, not to identify a perfect fit between a cultivar and an environment. And so that will involve understanding that no matter how well we define our TPE, there will be random variation in cultivar ranks within a trial series that can't be explained by factors or at least by environmental factors that you can predict before you plant the trial. A lot of environmental factors occur fairly randomly from location to location and from year to year. Yeah. So that's an important element, it seems to me, of this research, is to direct it to helping us with the real practical problem of determining really where the boundaries should be for our targeting. Once we've defined the margins or the boundaries of that breeding target, what we've got within the breeding target is essentially noise or factors that can't be predicted in advance of planting. So how do we deal with those? We need to think about perhaps their frequency so that we can weight them in parent selection. But you can't select a particular cultivar for a year that's at the high end of the cloudiness distribution as opposed to the low end of the cloudiness distribution. So, applying this to the problem of defining target populations of environments and then improving the weighting of information from trials within those TPE would seem to me a real contribution in terms of design of breeding programs. And I would encourage you to think about the design of breeding programs as your actual objective. This has been a problem with a great deal of GxE research. Plant breeders don't need a lot of help on defining or modeling GxE. They need a lot of help on defining what their breeding targets are.
Yeah, that's a good comment, Gary. Thanks. I think that helps all biometricians and QGs in the audience to and to question ourselves. I think even Pancho wants to immediately reply to that concern. Pancho, do you want to say something?
No. Thank you. Thank you, Daniel, for the good presentation. I think that yes, they use the factor analytic model. I mean, this is something that we are very familiar since the initial of AS Primal, even before the AS Primal was in place as a private, software. When AS Primal was public, we used to do a lot of factor analytic model because we think that this is a very parsimonious model that can help us a lot to assess GxE. My concern is in your presentation, do you think that is there any reason to come back to this sort of controversy between one stage and two and stage-wise analysis, after all the work done by, by different scientists at the level of well, of course, Brian Callis and Hans-Peter FIFO, where Hans-Peter shows basically always that the two-stage or stage-wise approach was in fact giving very similar results than the one stage and accounting very good for non-genetic variation. That may seem to me, maybe to me a sort of distraction issue that may not need a lot of extra work on that. That is my idea. We use basically for GxE and genomics, we use one stage when it's possible or two stage or stage-wise when it is possible. So that is one of my concerns.
Yeah, Daniel, do you have a take on that? And basically, can we do the exact same thing that you're proposing, but removing the part of, doing maybe the spatial analysis and the first stage within each environment, and then basically do exactly the same thing that you're proposing?
Well, I'm not advocating that, but also I'm not sure if I fully got your comment. Sorry, Pancho. But based on what Eduardo is just saying, look, yes, you can still do this in a two-stage framework. The reason why I'm not advocating, and yes, I'm aware of the extensive work of Hans-Peter in the area of stage-wise approaches and everything, and I don't think I have enough time to have a good discussion about this.
Yeah, just something brief.
Other than to say that I do take Brian's side on the one stage approach, but again, it does depend on the size of your data set. And more my comment was that depending on the size of your data set, you should be really sort of dictating whether you do a one stage or two stage approach. But I was a part of a team in Australia that were analysing national variety trials in December with a 30-day turnaround for something like 30 different METs, and we did it all with one stage approach, and they were not taking that long to run. In terms of the different weights and whatnot used in a two-stage, yes, if you end up using the full covariant, the full weights matrix, then you get quite similar results. But in the one stage, we are estimating everything from scratch, basically, taking start values and whatnot. But anyway, to sort of leave aside, yes, you can do what I'm proposing here in a two-stage approach. You can first adjust for your means if you wish, and just take through the means of pseudodata in a second stage. The covariates are built on the environment level, and then the genotype by environment interaction coincides with that as well.
I think that should suffice, Daniel. As Banjo mentioned, that's a controversial topic, but it's good to hear your perspective as well, and the one from Pancho, which I think is also pretty good and respectful.
Yeah, for sure. Thank you. Thank you, Dan.
Thanks, Pancho. Good comment and question. Valeria,
Yeah.
Probably you're going to follow up on Gary's comment.
Yeah, thank you very much. A wonderful presentation and wonderful approach. Actually, we were trying to do something similar. And I wanted to, I don't know whether I capture it properly in this slide, but I have the feeling that you are suggesting some of the components of environment which are related with, or correlated, better said, with some specific factors of the factor analytic, right?
Yep.
Yeah, okay. So
I guess you can interpret it somehow that way as well, right? Daniel? I mean, just to simplify it, I mean, what's behind each of the factors?
Yeah, so I mean, if I just talk about a conventional factor analytic model, again, I didn't want to put too much sort of theory behind it, but basically it is a bit like a multiple regression. You have these environmental loadings that would be your covariates, which I have here on the x-axis, And then you have your genotype scores, which are given by the slopes of these different regression lines. So essentially, it's a model that is saying that you have a regression of your genotype effects on your covariates. So in this case, the way that we not only estimate the loadings is usually estimated from the data, which sort of distinguishes it from a normal regression. In this case, the x-axis here is actually contributed by the covariates on the right-hand side there. Well, not those values, but the raw covariates as well that input them there. So you end up having a covariance structure that is basically the addition of a latent portion as well as a portion contributed by the covariates.
Right.
Yeah.
So yes, so at the light of what you just mentioned, then it comes to my mind that we will realize which are the main components of environment that are influencing the performance, and then we could somehow merge that into geographic information systems to identify potential regions where our materials could perform well, even before, let's say, sending them. And I mentioned that because at the light of a breeding program and how we designed it, for example, in the case, let's say a company is producing corn hybrids, but it's serving several regions. And actually, breeders used to interchange genotypes for the breeding programs. So it will be interesting to know whether a certain region in Europe or the performance in a certain region of Europe will correlate very well for a certain region in I don't know, China or North America. So I think if we are able to merge the information that you are providing here with geographic information systems, it will inform a lot on that. I don't know, it's more brainstorming.
No, that's good, Valeria.
Absolutely.
Yeah, no, that's good. That's good. I myself, I agree with that. I think we need to actually use this type of approaches to just define properly the TPE, put some boundaries, because once we know where the boundaries are, as you mentioned, Valeria, we can extrapolate those boundaries to different regions of the world.
Yeah, just before you go on, Eduardo, because you mentioned the TPE stuff twice now, and I'll just quickly go to this slide. What I really didn't sort of say was that I have partitioned these matrices based on north, south, and west, because this is the growing regions that we have in the US. What is a typical approach, or sorry, maybe I might not say typical, but what's a popular approach amongst plant breeders is to actually cluster these environments. Now, again, I'm not advocating clustering per se, and there's many sort of reasons why. One of them is because you don't really get much interpretation from what's driving the clusters. All I will say here is that in this case, you will, because you will get a certain number of covariates that are actually driving those different clusters. So again, like you're saying, you can start actually identifying the drivers for potential TPE identified by a cluster analysis.
Yep. No, good comment, Daniel. Thanks. Unfortunately, we hit the limit of the hour. So thanks for the good discussion. Thanks for the ones that actually wanted to throw some comments in the chat or also live with Daniel. I'm gonna, share the recording in case someone that had internet problems had to come back multiple times, as I noticed. And please, like I mentioned, if you want to share something with the group, send me an email. We'll be happy to have a discussion with you. This is a safe forum to share some ideas. Thanks again, Daniel. I think that was really interesting. I'm looking forward to reading that paper.
Yeah, thank you again for having me. And I think I'm just going to quickly copy these questions just in case, so I can answer them after the discussion or after the chat. So thank you again.
Sounds good. Sounds good. Then I let you go. Some people, is already leaving, probably because they have other meetings or they there is also some other webinars going on, I think, right after this one. So take care, everyone. Have a great day. Thanks, Daniel, again. We will talk soon. And yeah, cheers to everyone. Take care.
Thank you very much.
Bye-bye.
Bye then.